Hello. Today we're going to be showing you how to properly use a spectrophotometer. In this case, we use it right after our chromatography lab. If you don't know, that's the separation of a mixture's components based on the concept of how molecules interact with each other, otherwise known as molecular interactions. So in case you don't know, this lab involves a knowledge of the Beer-Lambert law. Now, I'll explain it to you right now. It won't make that much sense, but you will need to keep it in mind for later. Trust, this can be really important. So we're going to be using this kind of fancy scientific definition. It basically says that the absorbance of a solution is directly proportional to the concentration of the absorbent substance and the path length of the light through the solution. Now, the equation, I'm going to put it on the screen. A is the absorbance value, epsilon is the molar absorptivity, B is the path length, and C is the concentration. A word you finally recognize, right? Anyway, but trust, this will be useful later, so write it down. Anyway, we're going to move on and get into the lab. Okay, so as always, first things first is safety. As you can see, we have our proper lab attire, gloves, goggles, all of that. So in the second exercise of your chromatography lab, you're going to start getting your samples that you like to scan in the spectrophotometer into little cuvettes. There should be a total of 4 samples. One is going to be the blank, which has the deionized water, and then the other 3 colors that you get from the lab. Now, you may be asking why there's like a little blank cuvette. Trust, you'll find out soon. Just keep that in mind. Anyway, for the remaining 3, take a small portion using a pipette and fill the rest of it up with the mark, up to the mark with the deionized water. Once you have all 4 cuvettes, you're going to go to the spectrophotometer room to collect a UV visible spectrum that scans through each sample of your color collected. Now, once you get to your station, you will use the cuvettes with the colored solution along with the blank. Now, the reason we scan across 800 to 200 nanometers, which you will see later, is because we want to scan all possible wavelengths to find the one with the maximum absorption value. So once you get to your station, open the scan program and click select and record 800 to 200 nanometers. Now you're going to take your blank cuvette first, making sure you use a Kim wipe to wipe both sides, that the light will pass through, put it inside the spectrophotometer and close the lid. You're going to go to your computer and you're going to click 0 to zero out the deionized water, and all of our color samples we'll be using. Now, this is the part I told you to wait for. The reason we have the blank cuvette is to make sure that when we start scanning our other cuvettes, the absorbance value that the spectrophotometer is measuring is only for the color solutions. Because remember, we filled it up with water too. It's basically like a scale, like how you zero a scale to zero out the beaker's weight when we're trying to find the mass of a sample or a solution, except this is with a huge machine called the spectrophotometer. Now, once it zeros, take out the blank and start with one of your 3 colored solution cuvettes. Once again, wipe down the 2 sides that the light will pass through and place it inside, closing the lid afterwards. Hit start on the computer, name your file and sample. So, for example, if the solution is green, name it green or whatever you want. Afterwards, allow your spectrophotometer to collect its data. Repeat this process for the other cuvettes. And for each absorbance that the spectrophotometer records, make sure you write it down, because if you don't, you're kind of cooked for later. So when looking at a graph or when making your spectra, the X value should be your concentration, which is the molarity, and the Y value should be your absorbance value. Now, this relationship between absorbance and concentration is linear. This is where the Beer-Lambert law comes into play, especially the equation. It represents the relationship between these two. So this is what you should have been recording in your notebook this entire time. You'll compare the spectra that you measured using your collected data with the spectra for each dot at the end of your lab period. So the more data you collect, the more useful it will be later on. Now, when you're done with the spectrophotometer, make sure that you clean up your area. Same thing for your lab area that you use the first 2 exercises. The solvents that you use need to be poured into the waste containers in the fume hood that are in the back of the room, or depends on your classroom. And the solid waste, like the TLC plates, can go in the regular trash can, and the capillary tubes can go into the glass disposal bins. So here's our lab area all cleaned up. And you're going to see on the next slide, we recorded data all throughout our lab, and we found our RF values. And then on the next sheet that you're going to see is the spectrophotometer data sheet that we recorded throughout our whole time in the spectrophotometer room. As you can see, we wrote down the absorbance values for each cuvette, and we ended up making a graph off our predictions off that data table. And later on, you're going to match it with 2 solution spectras. So the more data that you record, the more helpful it'll be.